Good morning. Myself, Dr. Amandeep Singh, working as assistant professor in Khalsa College of Pharmacy. I have a total teaching experience of 12 years. Today, the topic that we are discussing in the class is pharmacognosy and its role in pharmacy and what are the thrust areas of its research. So, if we are discussing about the pharmacognosy, before that, we must have the information about the pharmacy background. So, for your all information, pharmacy is a branch of science which deals with the study of drugs and it relate the medical science with the chemistry. We have variety of different drugs in different dosage form. All these drugs that we obtain either from synthetic sources or natural resources. So whatsoever the drug we are using in the dosage form, actually it is a molecule, and that molecule will either be synthesized in the laboratory or it is obtained from the natural resources. So pharmacognosy is that branch of science which deals with the study of drugs obtained from nature or the drugs that we obtain from natural resources like plants, animals, etc. Whatsoever the drug we are using in different dosage form, either it may be used in the form of synthetic source or it may be used in the natural source. For example, we have different drugs that obtain from nature. For example, we have cinchona bark. Cinchona is a plant which is used as anti-malarial drug. The active constituent of cinchona, which is actually responsible for the anti-malarial effect, is quinine. So quinine is a drug. We are using quinine as starting material, and we are forming or preparing a dosage form using quinine as different dosage form like tablet or suspension, etc. So, pharmacognosy generally use a starting material in the form of crude drug. Crude drugs we are having of two types. First one is organized crude drug, and second is unorganized crude drug. The drug that have appropriate cellular arrangement and cellular structure is known as organized drug. Whereas the unorganized drug does not have any cellular arrangement, means they are devoid of having any cellular structure. For example, the leaf of a plant that is organized, the stem of the plant which is also a organized drug. In respect to this, the latex of the plant, the gums of the plant, the liquid solution obtained from the plant sources, all are unorganized drug. For example, honey. Honey is one of the example of unorganized drug which we obtain from animal resources. So now I will discuss about the relation between pharmacognosy in pharmacy. What is the relationship between pharmacognosy in pharmacy or drug discovery? As we have recently discussed that pharmacognosy generally define the drugs that we obtain from nature. Now, the drugs, whatsoever the drug that we have available in the market, it is available in dosage form. We have the different dosage form like tablets, capsules, suspensions, syrup, etc. All these dosage form were prepared by using a drug. Now, the transformation of a single molecule or drug into the dosage form is required the different transformation process. That transformation process, when specifically we are starting with the herbs, that transformation process is known as herbal drug technology. The technology that use, that we follow in processing the crude herb to the finished product. Okay, so now there are variety of different factors which affect the quality of our finished product. If we are discussing about the quality of our crude drug, there are variety of different factors like the identity of the crude drug, because there are variety of different plant species which are morphologically found to be similar, but due to some reasons we are not able to identify these drugs because most of them are displaying the similar morphological character. So, if we are taking wrongly any other plant species which is not therapeutically active, then obviously it may affect the quality or effectiveness of our finished product. So, we must have assured that whatsoever the drug we are using for the formulation of any finished product, it should be of good quality. It should be of genuine, authentic quality. So this is our first thrust area of research in pharmacognosy, that is identification of crude drug on the basis of different parameters. Now the question is, how could we identify any crude drug? We have variety of different factors, variety of different methods by which we can identify our crude drug. For example, first one is we can identify crude drug on the basis of their morphological features by considering the morphological aspects, morphological characters, organoleptic characters, we can confirm the authenticity or authentic nature of our drug. Now, the second method is DNA barcoding method. So, in DNA barcoding method, we are using DNA-based sequencing of nitrogen bases in any DNA of the plant species. By considering the DNA-based sequence, the sequence of nitrogen bases, we can confirm the identity of any crude drug. Next, we can also perform the identity of any crude drug by performing different tests like chemical test, physicochemical test, etc. Now, after confirming that whatsoever the drug we are using for the formulation of finished product, the drug is of good quality, drug is of authentic nature, then we will proceed further with its processing. So in the processing, we have to prepare the different formulations, different herbal formulations, like we are using herb in the form of solid dosage form, we are using herb in the form of liquid dosage form, or any other semi-solid formulation. But one of the major prospect or aspect related to the utilization of herb in finished product is herbal drug interaction. There may be a possibility that one herb of any plant material, one herb may interact with the biological effectiveness of another herb. Suppose we are preparing a finished product. We are preparing some herbal in dosage form, and in herbal dosage form we are using more than two herbal ingredients. Now, one herbal ingredient is having anti-diabetic effect, whereas the other ingredient that we are using in the formulation is having is promoting the blood sugar level. Now, if we are using both the drug simultaneously in any formulation, then obviously we are not getting any positive result with that drug substances. So I mean to say that there is a possibility that the herbs may interact with each other, and such kind of interactions when occur in our formulation, that interaction is known as herbal drug interaction. The second thing, under the considering the factors related to the quality of our finished product, is development of a new chemical entity. The new chemical entity that we are using, that we are processing from the herb, is one of the major prospect, one of the major factor related with the drugs utilization. So NCE, what is NCE? NCE is any of the active constituent that present in our plant material. That active constituent is actually responsible for displaying the biological effect of a drug. The biological effect of drug will be controlled by the content or the quantity of a new chemical entity or phytoconstituent in our drug. If the quantity of phytoactive constituent, phytochemical constituent in our drug is found to be below the acceptable limit, then our drug is not able to display the effect. We want to say that whatsoever the drug we are using, it should having the sufficient quantity of active constituent. If our drug does not have the adequate quantity of chemical substance, therapeutically active substance in our drug, then utilization of that drug is not found to be good. Means we are using those drug which is completely exhaustive. If the drug is not displaying any pharmacological effect, then using that drug in the formulation is of no use. So that's why this is the another factor that may be considered important while processing the formulation of formulation of finished product from the drug substances. The the next thrust area of our research is metabolomic study in pharmacognosy. So what we will do in metabolomic study, we have variety of different plant species which are displaying the different therapeutic effects, and that therapeutic effect is due to the presence of variety of different plant secondary metabolites. So plant secondary metabolites are those substances which are biologically present in plant species, and those molecules are actually responsible for providing protection against any kind of pest or any kind of infection in the plant body. If that biomolecule is having therapeutic effect against the pest, against the microbe, then the same substance or same biomolecule may able to display the similar role in human physiological system. So that's why we are able to we want to identify those plant secondary metabolites from the plant sources. By identifying the plant source, by identifying these secondary metabolites, we also come to know that from where it is developed in the plant body. Plant body ਦੇ ਅੰਦਰ ਇਹ ਜਿਹੜੇ secondary metabolites ਨੇ ਇਹ ਕਿਸ ਤਰੀਕੇ ਦੇ ਨਾਲ ਬਣ ਰਹੇ ਨੇ, how it is producing inside the plant body, and how it is biologically effective inside the plant body. So such study is known as metabolomic study, because these metabolites or these secondary metabolites are considered as the one of the byproduct of primary metabolism. So that's why by performing the metabolomic investigation, we come to know about the fat of these biomolecules inside the plant body. The next thrust of area of research in pharmacognosy is the interaction of primary metabolism and secondary metabolism. We have the primary metabolism process inside the plant body. Primary metabolism process generally generates such biomolecules which are playing significant role in the growth, reproduction of plant body. But the different metabolites that release from the secondary metabolism process or the primary metabolism process may participate in the production of secondary metabolites. So by identifying the relationship between primary metabolites and secondary metabolites, we come to know about the chemotaxonomy of the plant species. If we come to know about the chemotaxonomical character of those plant species, then we can easily identify our crude drug for the preparation of a finished product. Now, once we have prepared a finished product, the quality of the finished product is considered to be very important. Whatsoever the finished product we have prepared, the quality should be assessed as per the World Health Organization parameters, because we must assure that the drug or the finished product that we are going to prescribe to any patient, it should be safe, it should be stable, it should be quality checked properly, so that the patient will not suffer from the hazardous effect of such substances. So that's why we have to perform the different quality control test for finished product. So currently we have different parameters, we have different guidelines, which we are considering the World Health Organization guidelines or Ayush guidelines, etc. All these guidelines generally give us information related to the quality check of the drug substances. Currently we have different parameters. For example, we have morphological parameters, we have microscopical parameters, we have chemical parameters, physicochemical parameters, and last one is biological parameter. Considering all these parameters, or by performing the evaluation related to the different parameter aspect, we come to know about the quality of our drug substance. If the value of all these parameters found to be within the acceptable limit or following the standard parameters, then we come to know that the drug is of good quality. So we can proceed our finished product for the prescription to the patient. That's it.